Morning students, here is your Seema ma'am again. Now in the last session we have seen about the structure of stamen and the structure of microsporangium. Now we will see the next topic, that is the process of microsporogenesis. Now in simple terms, microsporogenesis can be defined as the process of forming of microspores from pollen mother cell by the process of meiosis is known as microsporogenesis. Now how this microsporogenesis happens? Now in the last session we have seen that whenever the anther is young, at the central portion of microsporangium, that is the, what is microsporangium? That is pollen sac. Means you can say the future microsporangium will be the pollen sac. So at the central position of this microsporangium, there occupies a mass of tissue that is known as homogeneous tissue and which is known as sporogenous mass of tissue. So at the time of microsporogenesis, what happens? The cells of this sporogenous tissue undergo meiotic division and they are forming microspore tetrads. Now, what are these microspore tetrads? Each of this microspore tetrad is known as the potential pollen or the pollen mother cell, and each one is capable of forming of pollen. Now, whenever the anther dehisces, what happens? This microsporangia, they dissociate from each other and finally the pollen grains are liberated from it. In TS section of the microsporangia, we can say that there are thousands of pollen grains are present or thousands of microspores are present which form the pollen grains and that are liberated whenever the anther matures and dehisces. During that the pollen grains are liberated. So this is all about the microsporogenesis. I hope students you might have understood what is microsporogenesis.